Good afternoon, everyone. I would like to acknowledge the contribution of each member of this project. Monica, which is with us presenting, Elena Rubino, Sahmin Kim, and Luke Flory. So if you run or are paying attention to the news, you will find easily information about invasive vines in different parts. I'm showing here in the lower and the bottom of this slide one piece that I found in a news outlet from Tennessee that reads, "Kill the kudzu, vine growing danger in Sevier County." Then on the left, you see a news from the local newspaper in Puerto Rico, and you see electricity line completely covered by vines. And very often you see this in the newspaper because some people argue that they contribute to the blackouts that we experience here in Puerto Rico. However, if you go to international newspapers like The Guardian, it's amazing that you'll find also news about vines in different parts of the world. In the left, you will find a clip from, I think it was Hungary or from Romania, the great abandonment, what happens to the natural world when people disappear. And part of the article describes how vines are taking over places that are being abandoned by people moving from the countryside to the cities. Then in the center, the Fijian island being strangled by vines. It's another example, in this case, in a tropical region of the world. And finally, on the right side, you see a house in Japan, an IKEA house, houses that are abandoned, that is being covered, starting to be covered by vines. So there are some common denominators among these cases. And just a quick read of this news, you will learn about the causes. So some people argue that, mention that there are intentional introduction. Some of the species involved in these situations were intentionally introduced to these places. You also learn about or associate, or the news talk about land abandonment. Others talk about natural disasters as possible causes of the proliferation of these species. And they also mentioned that these causes might be influencing disturbance regimes, that these vines are smothering plant canopies, that they're overtaking human infrastructure, but also there's a positive feedback where these disturbance regimes, smother plant canopies, and overtake human infrastructure are in turn influencing land abandonment and natural disasters. But if you read deeper into these news, you will learn that the proliferation of vines is also affecting the provision of services, both by nature and services provided by humans, like electricity, that they're influencing carbon sequestration, ecosystem productivity, that they're influencing regeneration and restoration programs, and very importantly, affecting the livelihoods of many people around the world. And this is happening not only in forests, but it's happening in natural areas, in fields, in right of ways and infrastructure. So it seems to be their common denominators. And this would be an amazing project to use content analyses to learn what is the people telling us about this problem. But this immediately begs the question about the scale of this problem. And we will argue that this is a multi-scale problem. So we learned from the news that many places around the world are experiencing a similar problem. So I would argue that this is a global problem. But of course, this is lived and felt at regional scales and at local scales. And this in turn will beg the question, what is local and what is regional? But I will not get into that at this moment. Another important thing that this problem, I think, brings is the multiple component that it has. And so we will argue that we should have a multi-perspective problem to deal with vine proliferation. And in the lower right, you will see a diagram illustrating this point, where we have biological drivers, we have biophysical drivers, and we have human drivers. So diversity, taxonomic diversity of vines, the diversity of host vine interactions, the diversity of functional groups are all contributors or should be taken into account if we want to learn about a problem that is local, regional, and global. On the other hand, when we talk about biophysical drivers, we're talking about climate, but also we're talking perhaps about more local conditions as those experienced by mountain regions, where rough topography creates this very heterogeneous landscapes. And finally, there's a strong human component to this, both as being impacted by this problem, but also perhaps contributing to this. And we will not expand on this today. So, to understand a problem that is local, regional, and perhaps global, we have to use approaches that are commensurate with the problem. On the left side, we have the use an approach, for example, which would be the use of remote sensing, in particular hyperspectral remote sensing, that can allow us to better nail down perhaps what species are involved, especially in regions that are very diverse, and also might help us understand what kind of function might be altering these proliferating vines. The other approach that we can take or that we can use together with remote sensing is the use of species distribution models that might help us understand what is happening or where these plants are occurring today and where they might be under uh, changing climates. So on the left, you see a map. The background represents a biophysical variable as temperature. And on the top, you find dots that represent the occurrences of tropical kudzu. On the right side, you see that we have a map where the colors represent the probability of occurrence of tropical kudzu in the island of Puerto Rico. And you will understand in a second why I'm mentioning this. So below on the left side, then, we have biotic variables, biophysical variables, and human variables that we can study at local, regional, and these are embedded into a global scale. So what are the challenges that we have to understand this problem at these scales? One, the databases. The databases provide information about the occurrences of the species, provide information about the biophysical variables that are used in these models, as Mona will talk about in a second. And then the other important thing here, the other challenge, is the scale. What is the scale of the imagery? How fine are the pixels to perhaps that would allow us to perhaps map the occurrence of these species? What is the scale of the biophysical variable that we can use to develop the species distribution maps? So, but before we continue, we have to learn a little bit about the biology. We have said that we need a multi-scale, multi-perspective approach to look at the proliferating vines, but also we need to learn a little bit about their biology. And this on the left, you find the cover of a book written by Charles Darwin about climbing plants. This was in 1865. And it's really interesting to see how these plants have been of interest to scientists and, of course, people interested in gardening and producing food for a long, long time. But one thing that I would like to highlight is that appears in the bottom, hence, no doubt, it arises that there are in all quarters of the world so many climbing plants belonging to so many different orders. So he is emphasizing the diversity of climbing plants. In part, he argues because climbing plants do not have to put a lot of energy into the production of wood to allow the plants to reach the light. Instead, they use different structures, trees, shrubs, or other vines to reach the light. So the host can be trees, but as we are learning here in Puerto Rico, they can also be climbers. So we find these associations of climbing plants, many different species together. And what I would argue is a facilitation process, some species facilitating other species to find the light that they're looking for. So what are the challenges here? So climbing plants are diverse. They have different preferences for hosts. They have different traits, different taxonomy. There are many different species, and they have many different uses. Also, when they by living in a close association with a host, this raises the question about how to model species that live in close association with other species. Another challenge is that the context where these vines are growing has changed. Perhaps 100 years ago, introducing a beautiful vine with beautiful flowers did not present any problem. But now, when conditions have changed, they might represent a problem. So the context, the environment is changing. And finally, the proliferation status is really interesting because some people talk about invasive species. Other people talk about non-native with high invasion risk. Here in Puerto Rico, we're using the term proliferating species because there is no official list. Who's listing the species is highly diverse, whether you find federal government, whether it's a state government or lower levels in government, and how they're being listed. So there's all this variety about vines that are proliferating. And so under this scenario, the question is who will be the next one? Who's most likely to move? Where are these vines going to move? And how will disturbance aid the spread of these species? So all these challenges led us to the next part of this talk, which is defragmented the knowledge landscape of climbing plants. And this is a metaphor for when one starts to look into vines their status in different parts of the United States, including Puerto Rico. And so one of the amazing things of the region where we're working is the strong biophysical variability that exists in terms of climate and other conditions. If you move from east to west, the area becomes drier. If you move from south to north, it becomes colder. And so this is an amazing scenario to understand who might be moving where or who might be deterred from moving. The other important thing about this region is that it has shared histories in terms of managing because of the institutions that are involved at the federal level and around plants. It also has a shared history of disturbance, a shared history of transportation and commerce. So these are all factors that are contributing or might be contributing to the proliferation of vines in this region, albeit with some differences. So how did we start defragmented the knowledge landscape here? So one of our first tasks was to create a list of climbing plants. And for that, we used the BONAP, the Biota of North America program. They have full listings of plants for the United States. And we added to that a list produced for Puerto Rico and the U.S. Virgin Islands by Pedro Acevedo. So this list comprises around 2,000 climbing species for the United States, Puerto Rico, and the U.S. Virgin Islands, and represents like the backbone of our work. Our second approach was to identify lists of invasive species of the different states and different levels of government. These are invasive species. Then the next step was to identify lists of noxious weeds, also from different agencies. And finally, we were able to obtain a list of plants for each state using a database called GIFT. So all these lists of plants went through the taxonomic name resolution service to homogenize the scientific names. And as a result of this, we generated a list of climbing plants with names that are used in larger databases that exist today. And then using that list and putting them together with the list of invasive species and noxious weeds, we were able to pull out the climbing species, both invasive and noxious, from the list that we had gathered before, and then also for the different states that we're including in this work. So now we have a homogenized list of climbing plants at different levels. And this led us to understand variation in the richness of these species. So if we start from left to right, we see Puerto Rico has 519 species, and the number in italics is the percentage represented by non-native species. Florida follows with 454 climbing plants, 39% of which are non-native. And if we go to the extreme, to Arkansas, we find 210 species, 31% being non-native. So there's a large variation in the number of species, including percentage of non-native species. And this begs the question, who will be the next one? Then also, if we put on top of this the number of species that are invasive or noxious, this is still in progress. We see that there is a large variation. For example, in Puerto Rico, 81 are listed as proliferating because there is no official list of invasive species in Puerto Rico, and 22 are listed as noxious weeds. If we go to Florida, 67 are invasive, and we have 26 that are noxious. And we have not gathered information for the other ones. So this is one way to understand the landscape of climber species in the United States and the territories here in the Caribbean. So the next approach would be to look at traits. And traits are characteristics of the plants that can have the same, can be recognized, let's say, carbon concentration on leaves. And it will be that currency is going to be the same anywhere where we can find them. But for now, I will not talk about that, but that's another possibility, and we can start defragmenting this landscape. So now we are going to talk a little bit about remote sensing and the multiple roles it can have to study vine proliferation. So the use of remote sensing to study invasive species is not new. Here I'm showing 3 different pieces of work that have used remote sensing to study invasive species in North Dakota, Tennessee, and Hawaii. And what I want to highlight here is that they use different platforms. That means satellite imagery, the 2 on the left, and airborne imagery on the right, and different sensors, multispectral data on the 2 on the left, and hyperspectral data on the right. And one of the tasks of this work is to detect invasive species. But actually using remote sensing can be more than this, as I will try to show here. So the idea is that remote spectroscopy uses the interactions between the light and leaves and canopy to characterize individual species or areas on the land that might have different properties. And from this imagery or spectra, as we see here on the left, each peak representing something different for each organism, we can obtain spectral features, and those spectral features can be used to perhaps identify species or map something that is called spectral species, or map something that is called functions that people working in agriculture are really using right now, for example, to look at the concentration of nitrogen in their fields, or concentration of carbon in their fields, or dry matter in their fields. So this is not new. People are using this actively at this moment, and we might be able to use these to understand the impact on invasive species. So, but this is generating a map of species, spectral species, or functions. But if we had images collected over time, this can allow monitoring species, that is the dynamics of these invasive species. So this is important because it will be similar to monitoring the spread of a disease. We can also use this information, if we were able to map species, for example, to inform species distribution models, provide occurrences that can be used in species distribution models, or to use this data to validate species distribution models. And finally, I think that is perhaps one of the most important things. When we have a map showing the distribution of species, we might be able to put this into a spatial context to understand the mechanisms driving invasive species and their impacts, including the role of climate, disturbance, and diversity. So here in Puerto Rico, we started to explore the use of remote high spectral imagery to study vine proliferation. A couple of years ago, when Dr. Bernard Hubbard from the USGS Mineral Resource Program contacted me because he was going to start a campaign to collect hyperspectral data here in Puerto Rico. And I had been studying the central area, studying vines in the central area of Puerto Rico and also landslides. So this became an interesting area for him too. So in this area, the USGS in the spring of 2024 collected airborne imagery at a 3-meter resolution, hyperspectral airborne imagery at a 3-meter resolution, and that is depicted by the green rectangles. But also superposed on this is a blue square that represents hyperspectral images collected from a satellite, the Prisma satellite that has a 30-meter resolution, and we're working with both sets of data. And a little rectangle magenta in the center represents the area where we have been testing a series of ideas regarding processing of these images and the collection of information, perhaps to identify species, among other things. And the points represent sites that we have visited during the last 8 months that serve as training points. So this data is amazing, but it also has there are a lot of challenges in terms of data processing. And I will just go quickly on the left to tell you a little bit about what is here in these images. You have each color or each layer here represents a wavelength. And the USGS data set has 594 wavelengths because it's a hyperspectral cube. So it's a huge, huge data set in terms of the physical size of the data. And so then each little square here represents a pixel, and each pixel has a value, a reflectance value, which is the amount of light that a leaf or a canopy is returning back and that is picked up by a sensor, in this case, that was flying in a little airplane that flew over Puerto Rico. The other important idea that I want to mention here is that the gray square on the bottom right is what is called a neighborhood or a community. And I'm mentioning this because one thing that we can do with hyperspectral data is actually envision communities of plants and calculate diversity indices, as we will see in a second. And that can be important perhaps to understand habitats that vines are preferring, proliferating vines. So then broadly, we went to the field, collected training points, and also used the images in our GIS Pro and another agency here in Puerto Rico to collect these training points. Pre-processing of these images includes both cutting the images, eliminating atmospheric problems with the images, eliminating clouds, water, shade, and soil, and then selecting spectral features that we're going to work with to identify plants if possible or other features of interest. So here on the left, we have a red RGB representation of the hypercube. It's as we see it, human see it is. And you will see magenta and cyan points that represent sites where the vines were present, the magenta ones, and the cyan, the ones were represented by other covers. And on the right, you see the spectra for these points. And the brown one or the orange one corresponds to vines. The green one corresponds to trees. And you can see that there's a separation between the spectra from vine sites and those for greens, and it's more pronounced in certain regions of the spectra. So this is the information that is actually used to do the mapping, for example, of vines. So I mentioned that the cube has 495 wavelengths or layers. And so this is very difficult to manage. So one way in which the dimension of the data set can be reduced is to generate indices that actually represent functions, things of interest, as for example, amount of dry matter, which is the one on the left. So here on the left, you see an index called CCI that represents the amount of carbon in the vegetation. Then NDNI is an index that represents the amount of nitrogen in the vegetation. CR SWIR has to do with water content in the soil and the vegetation. So instead of working with the whole data set, one can generate these new variables or features that actually represent a physical quantity that is meaningful in terms of ecosystem function. And then the other way to do this is to run a principal component analysis and extract the components that have the largest variability and see and use it as a feature of interest for what is coming next. So here in this slide, I'm showing a supervised classification of the images using two different methods. The important thing here on the left is that the red pixels, the yellow pixels represent, in principle, vine occurrences. There's some cleaning that has to be done here, but you will see that vines are pretty common in this region of Puerto Rico. And on the left is another classification method, and here the pixels that represent vines are in green. So we have potentially a way to map pixels that have vines, and here each pixel is 3 meters, so it's a very tiny part of the landscape. Then with this hyperspectral imagery, one can also calculate the Shannon index, diversity metrics, including Shannon and beta diversity. So on the left side, we have the Shannon index across the region. You will notice here that the pixels look bigger, and they are bigger because they're calculated in a neighborhood after processing the images and identifying something that is called spectral species using the entire data set. And then beta diversity is describing the turnover of spectral species in this entire landscape. And how can we use this information? So one thing that we can do is ask the question whether the presence of vines or vine patches is more common in areas of high diversity or low diversity. And I'm showing here on the right side one possibility where we see that there's a huge variation of where points that were not in vine patches occur versus those where vine patches occur, which is in green. And this work, we need to continue collecting more points to arrive at a conclusion. But the difference is not large, but the variability differs between points collected in areas where outside vine patches versus areas where there were vine patches. So what are the lessons that we learn? Is it possible to generate maps of species, in this case, tropical kudzu? Yes, very likely. Is it possible to monitor using remote sensing data these species over time? Perhaps using Sentinel, which has a smaller resolution than Landsat data. Can we use this information perhaps to feed species distribution models? Yes, very likely. And finally, can we use this information to look at mechanisms and impacts? And we would say yes, very likely. And our field trips during the last 8 months showed us how landslides that were triggered by Hurricane Maria in 2017 have been extensively colonized by vines, including kudzu. So with this, I'm going to stop sharing so that Mona can start talking about species distribution models.
Sorry, I forgot to unmute myself before starting the PowerPoint presentation. There we go. So you should be seeing my slides now. Okay, so we are going to get
We're
into
not seeing your slides, Mona.
Oh, okay. Let me try again. I did not click the share button. I did not confirm share. Should be able to see it now.
Yes, we can now. Thank you.
Okay, I'm gonna hide this. Okay, so Carla talked about hyperspectral remote sensing applications at this regional to local scale. So we're gonna scale up both spatially and temporally. So we're going to consider ecological niche modeling or species distribution modeling that is generally performed at large extents and using climate normals over several decades. So also a longer temporal scale than a narrow frame, a couple of years or years of remote sensing data. Okay, so just recapping what Carla mentioned at the beginning of the talk about ecological niche modeling, we use known records of species presence, and then we match those with digital maps of, in this case, climate variables or environmental variables in general. And then the statistical model, what the statistical model does is it looks for associations between climatic conditions or environmental conditions and presence, and creates a model of that abiotic niche for that species, which then that model is used to estimate potential distributions at the using the climatic data that we provided, or projects that model to additional climatic data that we provide, in this case, future climatic conditions. So with an ecological niche model that was trained with current climate data and current occurrences, we generate a potential distribution map of suitability for the species currently, and also a potential distribution map or climatic suitability for the species in the future, using forecasts of climate for near term, or in this case, in this presentation, I will show you just the first 3 decades from 2021 to 2040. But this is the principle for ecological niche model. Specifically for this project, we are dealing with 2 main challenges. One is, as Carla very well illustrated, a large number of species to investigate. So we can do something called, in invasive species world, called horizon scan, where we take into account what is known about species presence and climatic conditions or environmental conditions, and we estimate what could be the potential distribution of species that may not even be present currently in our area of interest. So we have a large number of species to investigate, and we are limited, or we have to acknowledge and deal with the fact that availability of presence records may not be that great, which I will illustrate next. So large number of species, how many species are we talking about? Well, if we take just the world checklist of vascular plants, 821 species have any life form that is associated with a climbing or a vine behavior. And so what we did for the modeling part of this project is to query the largest database of presence records, the GBIF database, which stands for Global Biodiversity Information Facility. You see the number of records in that screen grab for GBIF. They have billions of records for all kinds of taxa. But so we wanted to download and see what's available for any possible vines, climbers of any sort. And then we wanted to take into consideration the fact that we are going to match these records to climate data. And in this case, we have climate data from 1970s to 2000. And we have two spatial resolutions, two grain sizes of climate data, 1 kilometer and 4.5 kilometers. So that resolution comes into play when we want to work at a more regional to local scale. We want finer resolution climate data, not 4.5 kilometers. We want the pixel size to be smaller, and the smallest available for these averages is 1 kilometer. So how does that affect how we deal with our presence data? So here's just a screenshot of tiny example of GBIF records for a species of vine, which species I did not include, but one of those 800 and plus species. So what you notice hopefully is that this is a download from GBIF. What you notice is, first, we have records that don't have latitude and longitude. So we have a locality description where the specimen was collected or observed. It's a picture. In this case, it's a specimen looking at the years. And so we have a locality description, but there is no latitude and no longitude. So we cannot really pinpoint. We cannot put this specimen on a map, and we cannot associate it with climatic conditions. So that's problem number one. Records without latitude and longitude, we cannot use them in our models. Then there's the next crucial point. We think crucial point is how much uncertainty or how much noise is associated with these coordinates. So the next, the red arrow is for a record that has a locality description. It was collected in 1984, has a locality description, has latitude and longitude, but it also has that coordinate uncertainty in meters, that is 14,000 meters or 14 kilometers. So basically, the person who read the locality description, there's a whole protocol. We don't need to get into it, but there's a whole protocol of how to assign latitude and longitude to a locality description, and then assign the uncertainty associated with that locality description. So in this case, the locality description is not precise enough or detailed enough. It's along a river, near the county line. It's not very specific. So the latitude and longitude associated or assigned to this locality description has a large uncertainty of about 14 kilometers. Then the blue arrow shows a very specific locality description, a specimen collected in the 90s, latitude and longitude assigned. The specimen, since it's from the 90s, the specimen might even have GPS coordinates, which should lower the uncertainty. So if we take these two examples of records for a species, we are trying to match their location to climatic data for our niche modeling purpose. What we are looking at is variation. So the record that has a 14-kilometer error or uncertainty is shown that the uncertainty is that circle, the radius of that circle is 14 kilometers. So what we see is a lot of variation in precipitation of the wettest quarter in this case. So we don't know exactly where that specimen was collected or the observation was made. We are not sure what kind of precipitation of the wettest quarter, we associate to this the presence of this species. Now, the better result is when the uncertainty is very low. So in this case, it's below 1 kilometer. Our climate data is 1 by 1 kilometer. So the pixels, the blue and light brown pixels, represent the size of the climate data. So in this case, we are associating that 300 meters or so radius is specific enough that we can pinpoint a precipitation value without much error. Okay, so we know this is a fact. Everybody, most people hopefully know who do ecological niche modeling. What does it mean for our project? So we want to take into account, we call them restrictions or criteria. So our first criterion is the time frame of our climate data and matching that climate data time frame to the dates when these observations or specimens were collected for the species. And then we want to take into account the effect of the uncertainty of that location. We are not so sure where it is. Does it matter for our models? And then third criterion is modeling the potential distribution of these species at a finer resolution 1 kilometer versus a coarser resolution 4.5 kilometers. So I try to create a color scheme here to keep in mind when we go through a couple of examples. So we have the climate resolution in purple, then we have the time frame, occurrence time frame, and then we have the occurrence uncertainty in the other two colors. So the time frame, again, the climate data represents averages from 1970s to 1970 to 2000. We can expand a little bit. We can allow a little bit of error from 1965 to 2005, just to include a bit more records. There are more records in recent times than, so if we add 5 more years, we may get more records and improve our models. Okay, so downloading all the data from GBIF, what we observed was that depending on our criteria or restrictions, we will be able to work with 667 species versus 795 species. This is a very optimistic. These numbers are very optimistic because these numbers of species refer to having found that species in GBIF with at least one record. Now, when we model species, we need 20, 25 records. Obviously, we cannot do an ecological niche model with one record. So when we add this fourth criterion or restriction, the sample size, the number of species we can actually model drops to 447, for example, from 667 species. So we lose 1/3 of the species in general, depends on the resolution and the time frame. Okay, so to give you concrete examples, I just gave you an overview of what we can do, what kind of information is available at this global level, Global Biodiversity Information Facility database, how many species we may be able to model. But now let's look at specific cases. So I'm showing you 4 examples. What happens when we start imposing these restrictions or adding these criteria? So the top two examples, and I'm only focusing on one time frame from 1970 to 2000. So the most restrictive one, we are not allowing those extra 5 years on either side. Okay, so the top 2 species are examples of species where considering or not considering uncertainty doesn't change dramatically the sample size, the number of records we have available for modeling. The lower 2 examples, 2 species, shows dramatic differences. So if we decide not to take into account uncertainty, so without uncertainty, the number of records you can see on the screen jumps from 52 to 806 records, so a huge difference. what does it mean spatially? So just taking the last example, here's, at 1 kilometer resolution, we have 17 records for which we have the uncertainty, and the uncertainty is 1 kilometer or smaller. If we remove that criterion or that restriction, the number of records jumps to 1,300 records. But then when we look on the map, here's what we have. If we are very restrictive, very strict about occurrences we use, we have the purple records. Very few records. We are missing most of the distribution of the species, known distribution of the species. So if we are allowing records that don't have uncertainty calculated, or records that have higher than 1 kilometer uncertainty, our sample size increases dramatically. And I forgot to mention the top right corner shows the native distribution of the species and the invasive the non-native distribution of the species. So it's a Southeast Asian species found in 3 states on the mainland. Okay, so we have very different sample sizes. We're expecting changes in the model performance. I'm showing you 2 types of metrics of how good the models are, how well they perform. No need to worry about what they are, but for each species, we have 2 metrics. And then we have, again, the top 2, the top row is the species that don't have such a dramatic loss of data when we add the uncertainty, versus the 2 species. The lower row is represents the species that do have a huge difference when we decide to include records with uncertainty. So in green are indicators of good model performance, and in dark red indicators of poor model performance. So what we see, if we look at the top left corner, we have the species Cucurbita melopepo, for which only at 1 kilometer resolution with uncertainty, the model looks good. Once we add records once we remove the uncertainty filter, the sample, the data set increases, but also the model performance decreases. So this is problematic. We have situations where our decision of what to include and what not to include changes the model performance. Now the low row shows that regardless of how many records we include in the models perform very well. So this is good news for the species that have a huge difference in the sample size. However, when we look at the predictions, this is really bad. So this is one species, current potential distribution and future potential distribution, and the 4 types of data sets at 1 kilometer and 4.5, the 4 types of data sets with and without uncertainty, all over the place. So this means that our decision of what occurrences to include with uncertainty, without uncertainty, really changes dramatically the model, what the model deems as potentially suitable. So this is my take-home point: is we really need to be careful about the uncertainty, because it doesn't seem to affect the model performance, but the outputs, when we look at the projections, they are pretty different. So I'm out of time. I will stop sharing so that Carla can take us to the end of the presentation.
Carla, you're on mute.
Sorry for that. So far we have talked about biological and biophysical drivers of vine invasions, and we didn't mention the human component. But I want to share with you today, and I actually request your help in distributing the survey that we put out, which is Untangling Understanding Vine Proliferation in the Southeast Cask Region, a multi-stakeholder perspective. It's both in English and Spanish, and it will be very important for us to obtain information. Is not directed to managers, it's directed to your grandmothers, grandparents, uncles, everybody that is interested in contributing information about the proliferation of vines in this region. And so, what kind of products are we expecting to produce? A review paper. We have this list, a backbone of climbing species for the entire region. We might be able to produce a map of one species for central Puerto Rico from remote sensing data, and also a map showing where the species might be getting into a state forest. We were going to produce some species distribution models. We have interviewed farmers in Puerto Rico. Now we have the survey, and we hope that this information will help us move forward with understanding vine proliferation. And of course, we need to acknowledge many different people. Bernard Hubbard, who provided the USGS hyperspectral data, Jean-Baptiste Ferret, who has helped us understanding better the hyperspectral data, people of the Southeast Cask, students, panelists, and interviews that participated in the Invasive Vines Winter Institute in 2024 and 2025, and people here at UPR, students and faculty that have been invaluable with their help. And of course, we pull out resources from the projects that you see below to help students move along with this project. Finally, this is something that has come out from interviews with farmers. If I pull a vine in my farm, all the farm moves, just highlights the problem that vines represent here in the island as it is happening in the States. So with this, we conclude our presentation. If you have any questions, please feel free to ask.